Good afternoon, and thank you for joining today's IVM webinar. I'm Morgan from IVM Technology, and I'm delighted to welcome researchers from across India to today's session. This webinar will highlight how intravital microscopy enables researchers to move beyond static snapshots and directly visualize disease progression in living animal models. By capturing cellular and vascular dynamics in real time, longitudinal intravital imaging can provide deeper biological insight while reducing the need for animal sacrifice and improving experimental efficiency. Today, you will discover how intravital imaging is applied across a variety of research areas, including immune cell dynamics, lung imaging in sepsis models, fatty liver disease, and longitudinal brain imaging. Next, we are pleased to welcome Sarah Hall, Technical Application Scientist at IVM Technology. In this session titled Stop Sacrificing Model Organisms for Snapshots, See Disease in Real Time with Intravital Microscopy, she will demonstrate how real-time in vivo imaging can reveal dynamic biological processes that cannot be captured through conventional endpoint analysis. And please don't hesitate to submit your question in the Q&A box at any time during the presentation. We will do our best to answer as many as possible during the Q&A session at the end. And if you'd like to learn more about today's speakers or explore additional applications through our homepage, please check the Research tab at the bottom of your screen at any time during the webinar. Sarah, so please begin.
Okay. Thank you, Morgan, for welcoming words. So hello, everyone. Thank you for coming to today's webinar. I am Sarah, a technical application scientist at IVM Technology. In today's webinar, I will be introducing you intravital microscopy, which you can utilize to reduce the number of animals via in vivo imaging and observe your model in real time. So this is how today's webinar will follow. So I will go over intravital microscopy and imaging platform of IVM Technology first. Then I will provide applications of intravital microscopy. Today, I will show you the images of immune cells, the lung, brain, and liver, and which were obtained using intravital microscopy. And then end with the conclusion. So now let me introduce you to intravital microscopy and the features of imaging platform. So intravital microscope is an imaging technique for real-time observation. The cellular and tissue levels dynamics in living organism is allowed with this technique. So this technology allows to actually see the natural living environment compared to the traditional methods. So many of you know the traditional methods involve in vitro samples like the cell culture or the ex vivo samples, the fixed tissues. These methods hold limitations that it cannot conduct the dynamic studies. However, the intravital microscopy can overcome this limitation by observing what happens in natural living environment compared to traditional methods. So using IVM, we are able to see the movement and the interaction in cellular level inside of the living animals. So you might wonder why is imaging technique that allows the observation of cellular level in living organism is so important. So the in vivo imaging system, those are widely known are X-ray, the PT, CT, these types, and these are the whole body imaging in macroscopic scale. So the limitation is that it only provides overall status in organ and tissue level and does not provide the cellular level resolution because it is a whole body imaging. So compared to this, intravital microscopy offers high resolution and real-time images in microscopic scale. Because this method allows real-time imaging in microscopic scale, so dynamic studies dealing with the biological response from the drug and the behavior from the drug, and the time tracking in disease model are enabled. So further, microenvironment monitoring is allowed through the longitudinal imaging within the same living subject in different time points, improving the consistency and reliability. Unlike the traditional method, which requires the sacrifice of separate animals at each time point, it enables inter-animal variability with longitudinal imaging in the same animal. Not only that, the visualization of real-time and cellular dynamics of biological process offers deeper perspective in research. So this is IVM Technology's Intravital microscope. It is designed as all-in-one system for convenient operation for users. It is not only convenient in use, but as well as in installing and maintaining the system. So this simplicity but compact system will allow researchers to focus on experiments, imaging, and importantly, it does not require separate darkroom like the other microscopes. So our imaging platform is built for in vivo imaging in live animals by providing an ultra-fast polygon scanner and real-time temperature monitoring system, the motion compensation, and the AI-based denoising software. These features allow high-resolution imaging of organs in living animals. The reason for needing all these features is because they play a significant role in obtaining in vivo data in high resolution. So we have these features to ensure the observation of live animal images. Because the animals are alive, there's going to be a constant motion from respiration and cardiac activity. So this physiological activity gives difficulty in obtaining high quality and stable images. So therefore, IVM obtains motion compensation to apply during imaging. This feature corrects the movements and to obtain the stabilized images. In addition, we have AI denoiser to remove the noise from imaging at ultra high speed. This feature allows to obtain the clean image that can be used for accurate quantitative analysis. And last and most importantly, we have the real-time temperature monitoring system. This ensures to track animals' body temperature throughout long imaging session. The temperature not also can be tracked, but can be adjusted if needed. And this will help to minimize physiological variability, but as well as help in maintaining the ethical experimental environment. Now I will move on to the components of our platform. So in real-time observation of cellular dynamics, it is very difficult to capture biological process as cells like the red blood cells and immune cells move extremely fast and rapidly. So in order to capture these cellular events, IVM technology implemented an ultra-high-speed scanning system by utilizing a polygon scanner and galvanometric mirror. So the integration of these scanning systems enabled the visualization of rapid cell movements in real time. So on screen, this is the real-time imaging of the fast-flowing red blood cells in the beating heart. This was captured 50 frames per second at 512 by 512 resolution using IVM technology system. So as you can see, the visualization of cellular dynamics in high velocity was allowed. And you can see that these cells have the movement of these movements of these cells are captured. And this is motion compensation system that I mentioned earlier. So as mentioned, the physiological movements are unavoidable, especially in heart and lung imaging due to constant motion from heartbeat and respiration. So you can see that this captured image without the motion compensation is blurred and there is difficulty in knowing exact structure of the organ. However, if you look at the motion compensation applied image, the movements are corrected and the blood vessels are clear, the cellular signals are captured. So this enables the accurate observation of the organ. So we have applied a motion compensation algorithm in our imaging engine and effectively correct these movements and obtain high resolution and motion stabilized images. So another component I would like to explain is our AI denoiser software. Like I mentioned, the physiological movements are unavoidable when imaging live animals with respiration and heartbeat. So we have AI-based denoising algorithm that can remove the noise without interfering cellular environments. So compared to the raw video, AI denoised video has clear and defined structure of blood vessels, and this enables the identification of boundaries and helps to distinguish actual signal from the noise signal. So this will lead to more reliable analysis as it allows to track cellular dynamics precisely. And the difference between the motion compensation that I mentioned in the previous slide and the AI denoiser in this slide is that the motion compensation is applied while in imaging and is one of the features of our engine software. But AI denoising is applied to the images already taken because it works on separate software and you can post-process the obtained images separately. So our intravital microscopy has a wide range of chambers and windows that can be used to image different tissues and organs. So these chambers and windows provide short-term and long-term images in the same animal, resulting in more reliable data. And these are our various windows and chambers in different organs. We have customized devices for abdominal organs and for the heart, lung, brain, and more. So these are customized according to the physiological characteristics of the organ. And for longitudinal imaging of the abdominal organs, it's possible for approximately up to 4 weeks. And heart for lung, the in vivo imaging is feasible for 2 to 4 hours because the procedure for in vivo imaging requires opening the ribs. However, the long-term imaging for brain, however, is possible for more than a year and almost up to mouse lifetime when the surgery is performed with minimal inflammation and once the mouse is recovered. So now I'm going to move on to the application of the intravital microscopy. I'm going to show you some of the studies with the intravital microscopy used and using the chambers and the windows that I have just introduced. So first, I am going to show you the immune cells in skin and the lymph node. So here are some of the images of immune cells in skin. So here the imaging was performed on ear skin with the heating test system to monitor and adjust the temperature to obtain body temperature of the mouse underneath. And this image on the right is the image obtained from ear skin. So the mouse that was used here was LICEM GFP mouse, which express GFP in neutrophil and macrophage so that you can see these immune cells are in green and the blood vessels are in red. And this red was dyed using the anti-CD31 antibody. And even the movements of the immune cells were captured using this longitudinal imaging from the ear skin. And you can see the time points are located on the top. And this is another dynamic immune cell imaging. Like the last example, lice and GFP mouse was used and CARS protein was injected for this application. So CARS protein is secreted as signaling molecule and induces the immune system. And this example shows the immune cell recruitment when CARS protein was injected. So here the only signal from CARS protein is noticed after injected intradermally. And these images are taken after 3 hours of injection and 6 hours of injection. The immune cells have recruited to the injected spot. Also, the intensity of the immune cells are different. And obviously, the image taken 6 hours after the CARS injection has stronger intensity of signal compared to 3 hours. So the number of the neutrophil that has been recruited has increased over time. And this is another imaging of immune cells, but observed in lymph node instead of the skin. So the image on the right is taken up to 120 minutes. And here are the immune cells labeled. So B cell in green and T cell in red. So the arrows indicate where the D cells extravasate through the fibroblastic reticular cells of the lymph node. So this is the lymph node. And these cells are indicated in blue. And the movement of the cell is quick in this video, but there are different movements of immune cells captured. And you can see that the cells are coming in and out of the lymph node. And I also want to introduce you the dorsal skin chamber, which is optimized for monitoring microcirculation and microenvironment in cancer model, especially since cancer is heavily related to immunology. And this is one of the ways to monitor cellular dynamics in skin. So on the left is how it looks like under the microscope with the dorsal skin chamber, short in DSC, is applied on mouse. And we have the heating system that I mentioned before, and we have the chamber with the chamber holder for the imaging. And here in the middle, we have the compartments of the DSC, And the right is the final result of DSC applied on mouse. So this is the procedure in applying dorsal skin chamber on mouse. So here the hair of imaging area is removed using the removal cream because it's a nude mouse. We are not shaving, but we are only using the cream first. And the removal cream is going to be wiped off. After wiping it off, the skin on the back is going to be folded for the chamber to frame on it. And of course, we have to sanitize before holding the skin. Just like that, the skin is folded, and the chamber with the O-ring is going to be placed on the front, and the skin is going to be punctured to insert the screw. And this screw is going to secure the position on the top, just like that. Right now, we're puncturing the skin first, and then putting the screw. You can see the screw has been applied to the frame, and it is going in to the skin, just like that. And then the other frame is going to join on the back, and it's going to be fastened with the nut. So in this way, the skin is going to locate in between the frames, like a sandwich. And that is how you secure the skin first. Just like that. And then these frames are going to be stabilized with the surgery by surgerying the both sides of the skin, just like that. One by one. Then that's the result of the surgery. And then there's the other two sides at the bottom of the frame that is going to be punctured the same way as the first one. So it's going to do puncture, and the screw and the bolts going to be used to secure the frames and skin in a different place. Just like that, another screw is going in, and it's going to be fastened with the nut. Like that, and this will secure the frames and the skin together, so the skin doesn't move when the mouse moves as well. Like that, and that's the last screw. And then the skin of the imaging area will be removed just using the forceps and the scissors. It is very important to remove the skin of the one side only, not the opposite side. And after removing the skin, the membrane has to be removed and without the injury blood vessels. And right now, with the skin and the membrane successfully removed, the exposed area is getting filled with the sterile saline to prevent the tissue dehydration. Then this part is going to be covered with the glass. It's a cover glass to cover the chamber and the exposed area. And this area is secured using a C-ring right now. Like that. It is secure. And now it's ready for it to be imaged. And this is how it looks like under the microscope with the DSC holder using the securing the position. So here, the dorsal skin chamber was used to monitor the cancer development. The dorsal skin chamber is often used in cancer studies as there are many studies of drug efficacy in tissues conducted. And now this image on the right is the image from the breast cancer model. And this model was utilized to observe the delivery of nanoparticles in skin that longitudinal imaging was conducted. So the green is the cancer cell, and the blood vessel in blue, and the nanoparticles in red. So you can see that the signal of the nanoparticles increase as the time passed, and showing the nanoparticle delivery to triple negative breast cancer. So you can see that the red signal is shown as the time passes by. Next, I'm going to show you the intravital imaging of lung in sepsis model. So before diving into the example, I will explain the compartments. Those are used for lung imaging and the surgery as well. So these are the compartments used in lung imaging. So these are the devices: the vacuum-based imaging chamber, the tissue motion stabilizer, and the ventilator. So vacuum-based imaging chamber is what holds the lung while in imaging. And this chamber is connected to the tissue motion stabilizer, the TMS. So TMS is the device that stabilizes the surface of the lung on the chamber by negative pressure. And then finally, ventilator is what maintains the stable respiration of the mouse throughout the surgery and the imaging process. And this is the surgery part. And the surgery of the lung imaging is going to begin with the intubation. So intubation is important in keeping the respiration of the mouse. And this is this is how you intubate the mouse. And after intubation, the mouse is going to be placed. And here are the devices that I just mentioned, used for lung imaging. And previously, this is TMS, and this is a ventilator. After intubation, the mouse is going to be placed on the imaging stage and connected to the ventilator for the respiration. And on the imaging stage, the skin above the lung is going to be incised and held in place to be out of the way. The lung has to be exposed. And then the muscle right of the rib is removed. And then the rib bone right above the lung is removed as well. Right now, you can see on the screen. Like that, you can see that the lung is over there, right on the rib. And then the vacuum-based imaging chamber connected to the tissue motion stabilizer is going to hold the tissue in place. This is how you manage the negative pressure. And that's the chamber that I mentioned, just like that. You can see that the lung is sucked into the chamber using the negative pressure. Like that. And we have the cover glass that's placed on the top that's gonna cover the tissue. And that was the final result of the procedure of the lung imaging. And you get up to the microscopy using like that. And on the right, this is the image of the lung, and it's the final result of the surgical procedure. And this on the right is obtained from this setting would look similar to this image on the right. So the image on the left shows the closer look of the vacuum chamber imaging chamber. This is a slightly older version, but it works and maintains the same. Since the lung tissue is difficult to image due to respiration, TMS that I mentioned before maintains a negative pressure that will hold the lung tissue and allows to capture high quality images, just like on the image on the right. So the image on the right is from the sepsis mouse model. So this was induced by injecting LPS, and further, the FITC dextran was injected to show the blood flow, shown in green. And anti-Lysis-G was used to label neutrophil here, which is shown in red. And this is longitudinal imaging, and it was continuously taken image up to 10 minutes. And on the left is a snapshot of the longitudinal image from the last slide. As the time passes, you can see that the blood flow does not reach a certain area of the lung, indicating this is a dead space from lung sepsis. So there is also change in the neutrophil distribution as well. You can see that the neutrophils are more clustered around the dead space compared to the on time zero. And so these are more clustered around the dead space. And on the right, this is the imaging of the red blood cells flowing inside of the vessel. Because the red blood cells were labeled using the DID, and since the red blood cells were labeled, its signal can be tracked and its flow speed can be measured like that using the separate software. And the components that used in the lung imaging can be used for heart imaging as well. The reason why this vacuum-based chamber can be used in these two different organs is because vacuum-based chamber is designed to hold the dynamic and fast-moving organs. So obviously those are commonly heart and lung in animal and body. So I wanted to briefly show you the intravital imaging of the heart. So this is image from a fat accumulated mouse, and this was imaged using Z-stack imaging mode. So Z-stack imaging mode is going to come up later as well. But this allows to image from the surface of the heart and enables to obtain the images in depth like this. And in this image, the lipid is in white and the blood vessels in red and the immune cells are in green. So you can see that you can also mark the depth of the image here. And you can see that the lipid is located at the outer surface of the heart, and then the neutrophils and macrophages are located near the vessels inside of the heart. The next application is intravital imaging of liver disease model. So before I explain about the liver disease model, I want to show you the surgical procedure of applying abdominal imaging window. This window is one of the windows that allow the long term, and then the mouse can live up to 4 weeks, with the AIW attached through the surgery. So first, the hair of imaging area is removed using a clipper and removal cream. And after wiping off the removal cream, the skin is sanitized. And right under the ribs is incised after the sanitization. And then it's gonna incise a little bit to fit only up to the size that the window is gonna fit, not too much. And then around the incision, the purse string suture is going to be inserted around the incision area, just like that. And it's gonna create a loops. And about 3 to 5 loops. Suture is going to go through the both membrane and skin. Like that, you can see that these loops. And these loops are going to be used to hold the windows when it's inserted. The window is going to be inserted like that. Once the surgery is complete, the abdominal imaging window is placed on the incision with the membrane like that. And with the membrane and skin is going to stuck into the window. And this is going to be tightened and secured on the abdominal like that. Now the window is going to be held in place. And then the glass going to be attached on the top to cover the window, the exposed area. We use the instant adhesive to hold the glass. This is how you put the glass together. And this is the final image of how it looks like when the mouse is ready to be imaged using AIW. Now we will move on to the liver disease model that is imaged using AIW and our technology. So this is image from liver fibrosis. So this image was taken using a two-photon mode that fibrotic collagen was captured using second harmonic generation, SHG. So SHG is a method that can detect the non-linear structure without labeling with the antibodies, and it is shown in green here. And the lipid droplet is shown in pink. And one of the imaging functions available other than longitudinal imaging that I mentioned in the heart as well is the Z-stack. So this imaging was taken from the surface of the liver and then to the inside of the liver. So intravital imaging is also available to taken in depth, and how much it has entered in depth can be indicated on the top. You can see that the fibroses are more located on the outer surface, but also visible inside as well. Now, this is another model of liver disease. This one is the metabolic dysfunction associated steatotic liver disease, MSLD model. On the left, this is how you see here that the accumulation of the lipid droplets. So the lipid droplet is labeled by SF44, shown in yellow, and the liver sinusoid is labeled by anti-31, shown in blue. And you can see the accumulation of the liver tissue, how this disease model has fat built inside the tissue compared to the normal mouse model. And you can obtain the data in 3D image using the Z-stack mode. This is in 3D. And then the accumulation of the lipid was observed. So MSLD model was induced by MCD diet. And this MCD diet was monitored up to day 21 from day 0 from the normal diet. So you can see that the size of the lipid droplets in the liver becomes bigger as the time passes and MCD diet was treated. And this image on the top shows both the lipid droplet and liver sinusoid. And the image can be created, the channels can be separated, and the image can be created separately using the different channels. So therefore, the image only with the lipid droplet is obtainable because it was taken in different channel. But you can see that this image was taken together with the sinusoid and lipid, but lipid droplet can be separated and obtain the separate image. So one of the characteristics in MSLD model is that the lipid droplets are found far from the portal vein. So this could be observed using our mosaic imaging function. The mosaic function enables to obtain the image of larger area by stitching the multiple images. Those are next together. And you can see that this is the portal vein and the sinusoid is far from the portal vein. And then the lipid droplets are more observed around the sinusoid instead of the portal vein. And further, the size of the lipid droplet can be quantified to make the graph of it, or even the area of the droplet as well. So the separate image of droplet is going to be helpful in this quantification. And this is our final application, the longitudinal imaging of brain. So this is cranial window with the equipment to be used. And this with the cranial window applied on mouse, it can be used to monitor brain inflammation or the drug pharmacokinetics in brain for new drug development studies. And this looks like how under the microscope. So now I'll show you how this is prepared. So like the last videos, the hair of the imaging area is removed using clipper and the removal cream, and it's going to be applied and it's going to be wiped off. And of course, it's going to be sanitized. But before that, the mouse is placed on the stereotatic mount right now so that the head is fixed because the head is round, it can move, and so it does not move. So the place is going to be incised, the skin right above, and the skin, after cutting off the skin, the skull is going to be exposed like this. And then right now, skull is getting scraped by Mayer-Hoffer curette to mark the drilling point on the skull. That scraping. And then the skull is drilled using the hand drill. That right on the top. You have to be careful not to drill too deep or else it's going to hurt the brain. It's important to just drill only the skull. And then the bone flap of the skull is removed. Then using the forceps. And this 3mm cover glass is going to be used to cover the excised skull area. And in order to secure this cover glass, there are 3 parts to it. So first, the instant adhesive is going to be applied around the edge of the cover glass. And then the UV curable is going to be applied around the drill area as the second part in securing the cover glass and cure it for a minute or 2. That is curing. And once it's dry, the dental cement is going to be applied as the last part to secure the glass. It's going to cover over the skull after adhesive and the UV is dried. That's going around the skin. And we make sure that it doesn't cover the cover glass part. And this is it looks like after the surgery, and the cement is hardened. So using this method, the brain imaging can be conducted, and the mouse can live up to lifetime with this inflammation taken care. So this is one of the applications on brain, and this is from photothrombosis induced model. So using photothrombosis, the cerebral microinfarction model was induced by injecting the Rosebangle and irradiating the target area using the 561 nanometer laser. And you can compare the before and after induction of a thrombosis by using the DHE and PI staining, comparing it. And as you can see, the reactive oxygen species and the cell death was visualized comparing the successful photothrombosis. You can see the before and after. The blood vessels cannot be detected after the infraction induced. And this is image from the observation of clear scar after microinfarction induced. So with the cranial imaging placed, the longitudinal imaging was allowed, and the epithelial cells of blood vessels, the astrocytes, the pericytes are shown in blue, green, and red respectively. So, and the expansion of the shrinkage of the tissue was tracked at the vasculature map, and the area alteration could be tracked by the imaging each time point. And we can quantify this signal and turn into information into a graph. So when these numbers are quantified, the alteration of the area can be observed in numbers and clearly show the tissue has the expansion and shrinkage over time in microinfarction, not just only in images. And in this same application, not just the immune cells, but the blood flow was observed using the intravital imaging. So here the 3kDA dextran and 2mDA dextran was used to observe BBB dysfunction after cerebral microinfarction was induced. You can see that in these images, on these intravital images, that there are signals of these dextrans are noticed outside the vessels on day 3, indicating the loss of BBB function. But also that some of these vessels are recovered by day 5, and is saying that BBB is in function as well. And you can see that these signals can be turned into a graph because it's quantified, the intensity is quantified. So next is particularly focused on observation of the parasites. So in this image, the elongation of parasites were captured in the time point when the BBB is recovered, so between day 3 and day 5. So here the parasite is in red or the pink. You can see that how the parasites are extending in the blood vessels, which is shown in blue here. You can see that these are the time points, snapshots, and the expansion of the parasite. And not just the elongation, but the cellular division was also captured in the same time period when the parasites were elongated and BBB function was restored. So this division was observed to occur along the vascular walls after microinfarction. So the BBB function, the blood-brain barrier function, and the immune cells were observed in the microinfarction model. So how the cellular dynamics were observed could suggest that the parasites play a significant role in restoring BBB function. See that the division of the parasites, the nuclei are separated. And that's it for the applications, and I would like to conclude today's webinar. The intravital microscopy from IVM allows imaging more than just single time point imaging in different animals, as multiple time points can be imaged in a single animal. Not only that, we offer various windows and chambers in variety, and these are designed to observe the dynamic organs that it opens access to regions that were previously difficult to visualize in vivo. And in IVM's in vivo imaging system, the function to maintain live animals included to maintain body temperature and support physiological condition. With this system and the function ensures to obtain in vivo data and enable reliable imaging. And as I mentioned in the introduction, the traditional in vivo imaging method, the whole body imaging, results in low resolution data as the cellular dynamics cannot be captured, only in tissue level. But intravital imaging can overcome this limitation and enable to track the cellular dynamics, the minor drug delivery and distribution, and more, even in the organs those are challenging to observe, like the heart and lung, in living animals. And further, the IVM's intravital imaging system allows longitudinal imaging of the same subject over a long period of time, like you saw in the multiple times output in the application section that I have mentioned. And the traditional experiments usually require the sacrifice of separate animals at different time points, and the inter-animal variability has always been mentioned as a problem. However, with the intravital imaging system from the IVM, this problem can be resolved as longitudinal imaging within the single subject is allowed. So this will improve the consistency and the reliability in data. And real-time dynamic visualization of biological process in living animals will offer richer and more accurate perspective in complex physiology. So today I have showed you the different applications in different organs, the skin, the lymph node, the lung, liver, and brain. So I think you could see that the intravital imaging could be applied to all different spots easily, and that the intravital imaging is not only limited to one organ, or one disease model. It is a highly versatile platform that enables direct observation of cellular dynamics across a wide range of tissues and organs in vivo. So with these diverse intravital imaging platforms, we are able to capture the dynamic behaviors in cellular level and in real time in the cellular natural environment. Like the like you see on the screen, these are images obtained from intravital imaging. And traditional animal models have natural variability, meaning a massive cohort is needed to get a reliable data. However, with the intravital imaging that I have introduced today, the intravital microscopy can track the exact same animal over time and before and after the drug treatment and so on. So this can help in decreasing the inter-animal variability and significantly increase the reliability of the data. So that's it for the presentation, and thank you for your attention. So if you have any question, like Morgan said in the beginning, please leave it in the Q&A box. Thank you.
Thank you, Sarah, for the great presentation. So there's some questions over the Q&A box. And the first question is, hi, it was a nice talk. So in longitudinal imaging of the brain, what was the imaging technology used? What depth was achieved or possible with this technology?
That is usually allowed if you use a 2-photon, is usually up to more than 200 micrometer, but if you use a 2-photon, it up to 5 micrometer. I guess the technique that you're asking was that we use the 2-photon. Usually for the brain, we usually use the 2-photon to image the brain part because it goes in depth.
Thank you. And there's another question from the anonymous attendee. The very short question. So regarding the CD31 staining for the liver sinusoids, did you intravenously inject the CD31 antibody prior to imaging?
Yes, that's right. The CD31 was intravenously injected almost 1 hour before the imaging.
Yes, thank you. And the I think there's a long long question and chatting from the Q&A box, but I think we will address after the webinar. We'll address the email. I think maybe you are the user our IVM system, so it would be great that you can read the email address to chat or Q&A box. We can address your long question. So I think it's the all question in the Q&A box at the end. So yeah, thank you for everyone for your great questions. And for researcher in India who would like to learn more about our intravital microscopy solutions or schedule an online demo, please feel free to contact our local partner INCISE Healthcare at customercare@incisehealthcare.com. Their team will be happy to assist you with product information, application discussions, and demonstration requests. The webinar recording will soon be available on our YouTube channel, so if you have any additional questions or if we weren't able to address your inquiry during the Q&A session, please feel free to reach out to us at information@ivimtech.com. And we would also like to extend our sincere thanks to INCISE Healthcare for their support in organizing and promoting today's webinar. Thank you again for joining us today, and we look forward to seeing you at our next webinar. Thank you.